So I'm, like Aaron, I'm a postdoc in Carl's lab. Like Aaron, I did my PhD doing EPAs in behaving animals at MIT. I was doing tetrode recordings in Matt Wilson's lab from behaving rats. Also, like Aaron, I'm doing something different here, but a little bit less different. I haven't moved all the way over to the dark side of optical imaging and still sticking with the, the tried and true methods of extracellular EPAs. I've been doing a certain amount of work in integrating some of these techniques with optogenetic methods, along with lots of other labs, of course. And I think there are some special considerations. And again, of course, this could take a lot longer than 10 minutes, but I'll try to just sort of hit on some interesting topics for discussion. Very brief introduction to optogenetics. You hopefully have all seen these creatures before: channel rhodopsin, halo rhodopsin. They are light-gated or light-driven ion channels or ion pumps. And the reason why they are so exciting is that they operate, they allow you to control neural activity at native timescales of the circuit, so on the sort of millisecond timescale. They act through a physiological mechanism. That is, they directly control the membrane potential of the targeted neurons, and that allows the targeted cells to then recruit native circuits. Unlike, say, E-stim, electrical stimulation, they allow for really sophisticated targeting, pretty much anything you can come up with, whether it's genetic targeting, projection targeting, and then you can combine all those with an additional level of specificity introduced by where you put the light. And another nice feature of the methods is that they are compatible with animal behavior. Just like when you're doing EPAs in behaving animals, you basically just need a tether. And we all now have lots of experience having animals do, we as a field have lots of experience with behavior in tethered animals. It's not totally naturalistic, but there's lots of interesting things that tethered animals can do. The claim I would like to make is that optogenetics and extracellular EPAs are really highly complementary techniques. They go together well. EPAs can resolve the effects of optogenetic stimulation at that native time scale. So it's cool that you were able to have this sort of millisecond control of neural circuits. EPAs allows you to really watch what's going on. And in principle, we should be able to do optical stimulation as a sort of totally orthogonal channel. We should be able to record electrically and deliver light anywhere we like and not have to deal with electrical artifacts such as are inherent to electrical stimulation. And just like we can do behavior with tethered, with animals that are tethered for electrical recording, we can do the same limitations are in place for current methods of light delivery where we have a fiber optic cable coming to the animal. And as an example of cool new things you can do, we can use these great targeting techniques to measure neural activity and do online fast feedback using optical stimulation. Of course, you could do this with electrical stim as well, but all the advantages I mentioned before obtain. And this is a pretty nifty technique, which is still kind of getting the kinks worked out, but the idea of optical tagging is that if you're recording from a neuron, and this is a sort of a long-standing problem in the extracellular EPAs world, you'd like to know what type of neuron it is, or you'd like to know something about it, maybe where does it project to, or where does it project from. In principle, you should be able to, if you use your nifty targeting techniques to deliver opsin to a particular cell, go in and do your cell hunting experiment, just like you're used to doing. You find it, shine light, and watch and see whether that cell responds directly to light. We could talk about some of the reasons why that's hard. One issue is it's actually hard to tell whether a cell is being directly driven by light or is sort of being activated by something downstream. But this is more just meant as a, reasons to be excited.
Question?
Yeah.
You say this no artifact, recording artifact in principle,
Yes.
not in practice.
I'll get to that. In practice, basically, you can work around it, and it's not too bad.
No photoelectric effect?
Yeah, there is a photoelectric effect. I've got a slide on it, so we'll come to that. So I'm trying to limit myself to kind of things that are particular to this interaction between EPHIS and optogenetics, assuming that folks have, so light artifact falls in that category. Interactions between light, tissue, sort of tissue optics, and electrodes, and new devices. Aaron showed some nice pictures of devices. I'm going to show some nice pictures of devices with about the same level of discussion, but these ones have fibers integrated into them. And this, I think, is kind of I kind of wanted to make this my big point here, tweaking experimental design to manage complexity, and by manage complexity I mean keep things simple, and I'll hopefully make that point quickly soon too. So this is just for a little bit of kind of context setting. This is an experiment I ran where I tried to figure out how light was spreading within brain tissue. Brain tissue is highly scattering. Pretty much even if you've got a fiber in there with a very pencil thin beam, the light scatters very quickly. And the sorts of spatial scale on which you get light spread, I think this is like a 1% contour line of how bright it is right at the tip of the fiber. So light power is down by about 100x once you get a millimeter deep from the fiber. That's very much a spitball number, but it sort of tells you if you're going to put an electrode in and you're going to put a fiber in, roughly how close do you need them to be in order to get usable light intensity levels. So, you've definitely got a solid millimeter to work with if you want to be able to activate cells. And if it was really important to you, you could move that number out probably by a factor of 2 or more, either by choice of opsin or by pumping more light in or various other tricks. But that's the ballpark. Somehow a useful number for me to get my head around is this mean free path length of scattering in brain tissue, which is about 0.1 millimeters or 100 microns. And that means that on average in brain tissue, a photon will go 100 microns before it scatters. So if you're 300 or 400 microns away, that photon has undergone a scattering event 3 or 4 times. And I think that kind of helps explain why the light that's coming out of here isn't just going in this sort of straight cone, but it's a mix between a purely sort of isotropic spherical thing and a cone. So that's just a handy number. It doesn't really have much to do with EPHIS in particular. People often ask about heating, so I get out ahead of that one. Heating is not expected to be problematic. The kinds of changes we see are probably smaller than the normal changes you see in the brain with exercise or time of day, which can be like 3 or 4 degrees. And the other big confound to keep in mind is that light delivered to deep brain structures can almost certainly be seen by the animals. There was a paper out just recently from Anatole Kreitzer's lab at UCSF where they had the fiber was all shielded and they were seeing neural responses, reliably 40 milliseconds after they shone the light. And the only explanation was that it somehow was accessing the retina from behind and getting, they were getting neural responses in basal ganglia. So for all these reasons, you basically just have to use a non-opsin control. And this would be smart even if you didn't know that all these things were going on. But this is just more and more reasons. Just have a cohort of animals that have a non-functional opsin, same surgery, same virus, same light stimulation, because otherwise you're gonna confuse yourself. Never mind everyone who reads your papers. So after having said I was going to focus on EPHIS, these are not actually EPHIS specific issues, although potentially the matter of light being able to be visible to the animal is more of an issue with EPHIS, because you could see a real neural response that was time locked to light that was not mediated by any opsin. So the artifact is the light artifact at a metal electrode is for real. I think the sort of clearest demonstration that this is an effect at the interface of the metal electrode with the electrolyte comes from this paper by Jeff Carden from 2010. So they had a single fiber. I think this is actually done in saline, and they had both a sharp glass electrode and a metal electrode, and there's no effect in the glass electrode, and there's a very strong artifact in the metal electrode. Both a sort of fast time constant thing that's happening with every light pulse, and also a sort of buildup of charge across longer stimulation. So if you're using microwires or other metal electrodes, it's real. What we found in practice, I guess, is that pretty much it happens with a slow enough time constant that if you're already filtering out LFPs to look at spikes, you almost never see anything in the spike channel. Like if you look at, I've looked at lots of trains of pulses, it's really not an issue when you're looking at spikes, but it's a very real issue for looking at LFP.
But I mean, like this trace that we're showing here, it looks like a spike, right? I mean, it's
the time
that's
scale. This is 250 milliseconds. So if you look at it closely,
but I'm talking about the inset. So that's. I mean, if you have your filters at 250, you're going to call that a spike.
Well, I put them at say like 900.
Still, you're going to get something.
But it's. I'm just saying. Practically speaking, I rarely see even with like microwires really close to a fiber tip.
Okay.
I guess again.
Also had a paper saying it depends on the geometry of the tip. So this is actually one reason where smaller tips are better.
You would expect this to be proportional to the tip size.
Yeah, it's going to go down with surface area, and it's also going to depend on the orientation of the light relative to the fiber.
I was just thinking she's probably got it coming on sideways onto the electrode.
Whereas when it's straight above, you have less.
And it also seems to be material specific, probably solution specific. So it's real. This is yet another reason, I guess, to do your control animals where you don't have any opsin and you can see whether you're seeing an effect there. I've pretty much managed to convince myself that spike channels are pretty safe. And it really helps to convince yourself of that, to have an animal that doesn't have any opsin, where you also where you can't see anything in the spike channel, where the configurations are the same and the materials are the same and all the rest. Sure.
Sometimes a good comparison is to compare the waveform of spikes that you consider for your analysis before the stimulation, during the stimulation.
Yeah. I think that's a great idea. And depending on how you do your clustering, that might just fall out automatically.
Exactly.
You might see your clusters, there might be sort of a cluster here, and another one that's shifted over a little bit.
Visual thinking.
But your spike waveform could change for reasons besides having light artifact.
That's true as well, but at least first you have to see it, and then if there's a change,
you can assess it.
And the light artifact should be really sharply time-locked to the light, and it should happen with zero latency. So there are various ways that you would expect to be able to see a light artifact and know it and recognize it, and it should happen reliably. So if you can sort of filter in the spike channel and even like average on all your spikes, and you still can't see any kind of deflection that's happening in that high frequency band, then I think you can. I would say, I think we're fairly safe, at least for the sort of configurations I've been using, and you'd want to verify it in your own prep, but it's fairly easy to verify. The LFP is a bigger issue. These are, because these are like these weird buildup of charge at the interface, and I think this is sort of an engineering problem remaining to be solved. Ed Boyden's lab has some cool. They showed that by putting this transparent conductive coating on electrode sites, indium tin oxide, then they can almost get rid of the effect. So given that it is material specific, I think it sort of feels like an engineering problem rather than an inherent limitation of the technique. So the next thing that you need are devices that allow you to do simultaneous recording and optical stimulation. This is one from my old lab, Matt Wilson's lab, where they took the same sort of tetrode hyperdrive that Aaron was showing examples of and that you can buy commercially, and they just put a fixed fiber optic. They just sort of glued it in place with the fiber sticking out the end of the cannula at a known depth, and they never moved the fiber, and then they just moved their tetrodes normally in this sort of field of light illumination. This is the Optetrode, which Paulina and Keva, who was in Carl's lab, and Aaron published this year. It was just this year. It was last year. So this is this kind of stripped down, simplest possible movable electrode, movable fiber electrode setup as you can get. These are quite quick to fabricate, assuming you have the parts. And the configuration here is the fiber is movable, and it has tetrodes glued to the side of it at a sort of known depth sticking out from the end of the fiber, and then that whole thing is just moved as a single shank. And they implanted that successfully on mice and were able to isolate single units. Here's a case where I think you've got a pretty big probe going into the brain. There's no reason you couldn't make it with a smaller fiber, but whatever you do, it's still a pretty big thing to be shoving down there. And you don't have any lateral movement. So these are kind of one-shot experiments. You get to go down and hunt for cells on your way down. I guess you could, conceivably get cells and hold them, but I think because the whole shaft is so rigid, you can't hold cells for very long. So that's, every technique has its trade-offs. Tetrodes are nice in that once you get them in place, they tend to ride with the brain. So this would give you kind of chronic recording, not a movable fiber, and lots of tetrodes. This would give you a good chance of getting 1 or 2 cells, being able to isolate them, but it'd be more of a cell hunting experiment across a single recording session. And this is another compromise, which is a device that I came up with for a paper, a project with Ofer Yajar, who was a postdoc in the lab. And this is going back to what I said when I wanted to make the main point, which is that sometimes you really just want to make the EPhys, and in particular the EPhys plus the optogenetics, as simple as possible. So this is totally fixed electrode, totally fixed fiber. This is my thumb. This is a connector with a fiber ferrule glued to the side of it, and we just have a few microwire electrodes that are cut off blunt, glued to the side of the fiber, and staggered in depth. So these are maybe every few hundred microns. So you're sampling across a range of depths in front of the fiber. The whole thing is just implanted. It's not really any more difficult to implant than a single fiber would be. So it's a straightforward surgery. You can make lots of these, so you can do larger cohorts if you want to do it. If you're trying to do this in conjunction with a behavioral experiment, where say you need multiple animals.
And what was the impedance of these electrodes? They're relatively
these
low?
are big. These are 50 micron, so you're only getting LFP plus multi-unit, undifferentiated multi-unit, and I think sub 100 kilo ohm.
Good.
So you're definitely pretty immune to noise, but you're not resolving single units with any fidelity. But that's sort of not the point, So this is a range of tools. The other thing is that if you want to actually, there are lots of classes of optogenetic experiments where you might want to do your light stim in one area and your recording somewhere else. If you wanted to, if you had some area that was projecting broadly across cortex, say, you might, shove your fiber over that nucleus and do your recordings wherever you like. And in that case, just use whatever EPhys technique you're comfortable with, and you can shove a fiber somewhere else. You can do sort of multiple implants on the same animal. So here's my attempt at some humor. This is. I don't have a lot of data points that I'm fitting to this, but there's probably some room here. The key is that that power. I figured it out, because this is like, if you just walk in and you don't have to collect any data, someone just hands you like a data set, then that's 0, so 2 years. And if you're just trying to get one thing working, then maybe like 3.5, and you're trying to get 2 things working in the same animal every time, you're up closer to 5. So. There's probably a fudge factor there for how many happy hours you go to, how many kids you have. But the general principle is that the more things have to work within a single animal, the worse your life is going to be. And you made this point in your opening remarks, I believe, in terms of complexity. So here in blue are sort of new things that you have to add on to your already complex behaving animal EPhys experiment. So not only do your animals have to behave, and they have to be trained, and they have to stay healthy and not die, not only does the rest of your rig have to be working and you have to troubleshoot noise, not only do your recordings have to be good and stable across time, not only does your implant have to not fall off the animal, and your surgery has to go well, and you have to target everything to the right place, but your light has to be going to the right place as well. So you've got that complexity, and you've got to worry about viral expression and targeting of the virus, and whether or not the virus is, if you're using a virus or a transgenic animal. So you're really layering on these extra levels of complexity. I don't know how many of these count as independent techniques for the purposes of the equation.
You're going to be like 15 years.
Yeah, it's going to be a while. But to me, this is a really strong argument for, and I think one piece of evidence here is that a lot of the published papers are not doing, they're not doing, say, Wilson Lab or Bujaki Lab style recordings from huge populations of cells while they're also getting optogenetics working. The stuff that's coming out is either sort of simple optical tagging experiments, or it's LFP. They've got a behavioral effect, and the electrophysiology is kind of bolted onto the side as like, here's a nifty LFP effect. So arguably that's what Ofer, the paper I mentioned there, Ofer was. They had this really nice sort of story about the effect of light on the animal's behavior, so they could, they had a whole rescue type situation, and they said, well, let's add in some LFP. So we built these devices just to record LFP during a behavior that we'd already established was working well with the light effect. And I think that pattern is instructive anyways. So to bring it all together with an example, this is a result that just came out recently. Jeanne Paz in the Huguenard lab was the first author, and I was the next author, and she had developed this cool, again, she had developed a really sort of like solid biological result. She adapted this whole seizure model to induce seizures in rats, and it established that it involved the thalamocortical circuit, and had done lots of great in vivo physiology work on that. And then we said, okay, well, let's layer on this optogenetic experiment. In this case, it's arguably less bolted on. I mean, it was kind of instructive, but I think that optogenetics wouldn't have stood on its own without that really kind of solid, just straight E-phase result. So we took as a chronic multi-site optrode, implanted that into the thalamus. So here's the sort of overview of the experiment. So we had our fiber and our depth electrodes in thalamus. We had skull screw electrodes, which, if you remember from Aaron's talk, are just big fat stainless steel screws, which just give us EEG and don't even require a preamplifier. And then also up here I had the sort of time frame of the whole experiment, just to give a sense of how these things can work. She did the injury, which induced the stroke at time 0. A week later, did a virus injection surgery. 2 weeks later, implanted the devices. And then starting at 4 weeks, so about 3 weeks after the virus injection, we started being able to do the recordings. And those continued on for as long as the animal stayed alive. And so, to kind of give some examples of what we're talking about, we didn't try to get LFP from our thalamic electrodes, but we did look at multi-unit activity. And we found that when the animal was having a seizure, as measured by skull screw electrodes out here in the cortex, we could see bursts of multi-unit activity that were synchronized with that in the thalamus. And then we were recording those and looking for seizures, and then when we found one in the cortical electrode, we could go in and deliver light to thalamus and disrupt the seizures. So the kind of the point I want to make from this is that we're working around the issues of complexity inherent in tying all these tools together, using the sort of dumbest possible optogenetic tool, or dumbest possible probe. And even then, the wheels barely stayed on, right? You still only had 1 or 2 animals that made it through the whole gamut where everything was working well. But those sort of 1 or 2 examples, I should be more explicit, shouldn't I, whether it was 1 or 2? Let's say it was 2. There's a big difference between 1 and 2. That result sort of stood on its own a lot more strongly because there was a sort of, because there was all this other.
Did you do some kind of calculation to decide that you needed 4 different sites to get the whole thalamus? Or
What was the I think This was actually more about our uncertainty about where our virus actually ended up. We wanted to make sure that we had. No, you know what? So after the injury to the cortex, there's a part of the thalamus, the corresponding portion of thalamus dies off. So we knew that we had one part of thalamus that was should be dead quiet. We should have a neighboring area where we had virus, and we were just trying to hedge our bets in terms of covering a big, millimeter long chunk, and while keeping the channel count low for the same sort of complexity. So this was sort of all about designing for failure, and we knew we only needed one channel to work, and pretty much that's how it got. Okay, and I think that's the end. So future directions, reducing artifact would be great. I think it's probably going to happen. Things I didn't talk about at all: spatially modulated light delivery. If you have multiple fibers, or cool endoscopes where you can project patterns of light. That's unexplored, so far unexploited possibility with optogenetics and E-phase. And untethered, we already have wireless E-phase. You can go and buy these things. And I think there are folks who are working on wireless light delivery as well, though that's its own set of challenges. And then there's this question of whether it'll be made obsolete by GCamp 12, or whatever it is. Maybe Jesse would like to speak to that. Okay, thanks.